Our rods have the photopigment rhodopsin, and our cones can have one of 3 different photopigments. Those photopigments are responsive to a more narrow range of wavelengths. We have a blue cone opsin that responds to blue wavelengths of light. We have a green cone opsin that responds to green wavelengths of light, and a red cone opsin which responds to red wavelengths of light. So if blue light hits the blue cone, the photopigment will split, that is, the opsin will split from the retinol. It will initiate a signal transduction cascade to decrease the amount of cyclic GMP in the cell, and the cation channels will close, causing a hyperpolarization called the receptor potential. And the blue cone will stop releasing glutamate. If we have green wavelengths of light, those will stimulate the photopigment in the green cone. The green cone opsin will split from the retinol, initiate a signal transduction cascade, decreasing cyclic GMP, closing cation channels, causing a hyperpolarization called the receptor potential, and the green cone will stop releasing glutamate. Same thing will happen with red light and the red cone, causing a hyperpolarization called the receptor potential. Now let's look at what happens to the downstream cells. The red cone, via a bipolar cell, connects to a type P retinal ganglion cell, that is a parvocellular retinal ganglion cell, which is called the red-green retinal ganglion cell. That's because it gets information not just from red cones, but also from green cones. So this is the red-green retinal ganglion cell. Okay, and there's its axon. The red-green retinal ganglion cell has a baseline firing rate. When red light is present, the firing rate of the red-green ganglion cell increases. However, when green light is present, the firing rate of the red-green ganglion cell decreases. This is called opponent process vision. Our retinal ganglion cells use this rate code to tell us if we're seeing red or green. Now let's talk about how it's possible for the red cell to excite and the green cell to inhibit the red-green retinal ganglion cell. The difference is based on which type of bipolar cell the cone connects with. The red cone, via an on bipolar cell, means that the on bipolar cell is going to be in a relatively depolarized state when red light is present, and therefore it's going to be releasing glutamate onto the red-green retinal ganglion cell, where it will bind to AMPA receptors and cause an EPSP, increasing the firing rate of the red-green ganglion cell. In contrast, the green cone connects to an off bipolar cell. This bipolar cell will be off when green light is on. It won't be releasing glutamate, which means that AMPA receptors here will not be active, will be in a relatively hyperpolarized state, and therefore it will decrease the firing rate of the red-green ganglion cell. If we look at the blue cone, when blue light is present, the blue cone synapses with an off bipolar cell onto its downstream retinal ganglion cell. This is also, of course, type P, parvocellular retinal ganglion cell. And this retinal ganglion cell is called the blue-yellow retinal ganglion cell because it responds to blue and yellow light. It has a baseline firing rate, and when blue light is present, its firing rate decreases. Now let's talk about what happens in yellow light. We don't have a yellow cone. At the level of the cones, we have what's called trichromatic vision. Three different types of cone opsins. Each cone has either blue, green, or red opsin. So what about yellow light? Well, yellow light is sort of in between the wavelengths of green and red. So yellow light will actually stimulate both the green and the red cone. The green cone will inhibit, and the red cone will excite the red-green retinal ganglion cell. So when yellow light is present, the inhibition and the excitation cancel each other out, and we're back to the baseline firing rate. However, these cells can also excite the blue-yellow retinal ganglion cell, causing an EPSP, which will increase the firing rate of the blue-yellow retinal ganglion cell. Again, this is opponent process vision. We see either blue or yellow. This rate code where the rate of firing conveys information tells us either we're seeing yellow or blue. In the red-green retinal ganglion cell, an increase in firing tells us we're seeing red, a decrease tells us we're seeing green.